Introduction. For decades, the genetic history of the Yoruba people, one of West Africa's largest ethnic groups with over 40 million members spread across Nigeria, Benin, Togo, and the diaspora worldwide, has intrigued scientists and anthropologists. Recent breakthroughs in genomic research have finally unraveled long-standing mysteries about their ancestry, migration patterns, and the complex genetic landscape that defines this ancient civilization. These discoveries not only illuminate the Yoruba's past, but also reshape our understanding of human migration patterns across Africa and the world. Who are the Yoruba? The Yoruba people constitute one of Africa's most influential civilizations, with a rich cultural, linguistic, and historical heritage spanning thousands of years. They occupy southwestern Nigeria and parts of neighboring countries, primarily concentrated in regions like Lagos, Ibadan, Osun, and Oyo states. The Yoruba civilization is renowned for its sophisticated political systems, artistic traditions, and complex spiritual practices that have significantly influenced not only African culture, but also shaped diaspora communities in the Americas, Caribbean, and beyond. Historically, the Yoruba established powerful city-states, including Ife, Oyo, and Benin, which developed complex governmental structures, advanced metallurgy, and thriving trade networks centuries before European contact. Their descendants maintain strong cultural continuity, preserving ancestral traditions, languages, and social structures, even after centuries of slavery, colonialism, and globalization. The genetic questions. Scientists have long puzzled over several key questions regarding Yoruba ancestry. 1. Ancient population origins. Where did the Yoruba's ancestors originate, and when did they arrive in present-day West Africa? What were the primary migration routes? 2. Genetic ancestry components. What is the precise genetic composition of the Yoruba, and how does it compare to neighboring populations? Were there multiple migration waves, or a single founding population? 3. The Saharan expansion. Question, did the Yoruba, like many West African groups, experience northward expansion into Saharan regions during wetter climatic periods? And was there back migration from the Sahara? 4. Demographic history. What population size changes occurred historically? Did the Yoruba experience bottlenecks or rapid expansions? 5. Admixture events. Beyond the primary ancestry, were there significant admixture events with neighboring populations that shaped Yoruba genetics? 6. Comparison with global populations. How genetically distinct are the Yoruba from other human populations globally? The research breakthrough. A major turning point came with large-scale genomic studies published in recent years, particularly involving research from institutions across Africa, Europe, and North America. These studies employed several methodological innovations. Next-generation sequencing technology. Modern whole genome sequencing allowed researchers to examine millions of genetic markers across large Yoruba populations with unprecedented precision. Unlike earlier microarray technology that examined only thousands of markers, these comprehensive approaches revealed subtle population structures and ancient admixture events invisible to previous methods. Large sample sizes. Earlier studies often worked with limited sample sizes from specific Yoruba communities. Recent research expanded to include hundreds of individuals from diverse Yoruba settlements, providing a more representative genetic picture of the entire population. Ancient DNA integration researchers incorporated ancient DNA from archaeological samples across West Africa, providing temporal depth to genetic analyses. By comparing ancient genomes to modern Yoruba, scientists could literally trace how populations changed over millennia. Statistical innovations. Advanced statistical methods, including admixture analysis, genome-wide association studies, GWAS, and machine learning approaches, allowed researchers to disentangle complex ancestry signals and identify previously hidden population components. Key findings. The genetic story unfolds. Primary ancestry, the West African component. The research confirmed that the primary ancestry of the Yoruba originates from West African populations that have inhabited the region for thousands of years. This ancestry is distinctly African, with deep roots in the Sahel and Guinea savanna regions. The genetic evidence suggests that Yoruba ancestors were part of broader West African populations that developed complex societies in the Niger River Valley and surrounding regions. The strength of this West African component, comprising approximately 80–90% of Yoruba ancestry in most individuals, establishes the Yoruba as indigenous to their region, with genetic continuity stretching back at least 5,000 years based on divergent dates estimated from genetic data. The Saharan migration signature. One surprising finding involved evidence of historical population movement into and out of the Sahara region. During the African humid period, approximately 9,000 to 5,000 years ago, the Sahara received sufficient rainfall to support pastoralist populations. Genetic evidence suggests that some West African populations, potentially including Yoruba ancestors, expanded northward during this period. When the Sahara entered its current hyperarid phase around 5,000 years ago, populations retreated southward in a process called desiccation-induced migration. Genetic signatures of this movement, detected through admixture patterns in ancient DNA comparisons, indicate gene flow between Saharan and sub-Saharan populations during this critical transition period. This discovery solved a long-standing mystery. How much genetic exchange occurred between Saharan and sub-Saharan West African populations? The answer? Considerably more than previously thought, though these admixture events were ancient and relatively small in scale. Subsidiary ancestry, the northern component. Beyond the primary West African ancestry, genomic analysis revealed approximately 10 to 15% admixture from northern sources in Yoruba genomes. This northern ancestry shows affinities to populations from the Sahel region and possibly North Africa, consistent with historical trade networks and population movements across the trans-Saharan routes that flourished in historical times. This component is not uniformly distributed. Some Yoruba individuals carry more of this northern ancestry, likely reflecting more recent admixture events during the period of trans-Saharan trade, approximately 100 to 500 years ago, when merchants, scholars, and soldiers traversed these routes regularly. Internal population structure. Surprisingly, genetic analysis revealed significant internal structure within Yoruba populations. Different Yoruba communities, separated by distances as small as 50, 100 kilometers, showed measurable genetic differentiation. This microstructure reflects historical settlement patterns, different founding populations with slightly different genetic compositions, restricted gene flow, geographic and cultural boundaries that limited intermarriage between communities, temporal variation, more recent migrations, created pockets of distinct ancestry social organization. The Yoruba clan system and patrilineal patrilocal residence patterns influenced genetic distribution. This finding challenged the assumption that the Yoruba represented a homogeneous genetic population, instead revealing them as a collection of related but genetically distinguishable communities, a pattern common among ethnically and linguistically defined groups. The Ife dispersal model. Combining historical, archaeological, and genetic evidence, researchers proposed the Ife dispersal model as the best explanation for Yoruba peopling and expansion. According to this model, ancient period, before 2000 BCE, Proto-Yoruba populations inhabited the Niger River Valley and forest regions of West Africa, Ife period, 2000-1000 BCE. A cultural and political center emerged at Ife, which served as a demographic and cultural nucleus. Dispersal phase, 1000 BCE 1000 CE. Expansion from Ife resulted in the settlement of other regions, with groups carrying genetic markers traceable to the Ife source population state formation, 1000 to 1500 CE. Establishment of powerful city-states, including Oyo, Benin, and others, each with their own political structures, but shared Yoruba cultural and linguistic features. Historical period, 1500 CE present. Complex interactions with external powers, including trans-Saharan traders, European slavers, and colonial forces. Genetic evidence supports this narrative through the detection of isolation by distance patterns and the identification of shared genetic markers across dispersed Yoruba populations that are rare or absent in neighboring groups, solving the admixture puzzle. One of the most significant breakthroughs involved understanding admixture timing and sources. Using sophisticated dating methods that analyze the length of DNA segments inherited from different ancestry sources, researchers determined ancient admixture 3,000 years ago. Genetic evidence of mixture with Saharan populations during the humid period represents the oldest detectable admixture event. Historical admixture, 500-1500 CE, trans-Saharan trade routes brought individuals from North Africa and the Sahel into contact with West African populations, resulting in detectable gene flow visible as shorter admixture blocks in contemporary Yoruba genomes. Transatlantic slave trade effects, 1500-1900 CE. While enslaved Yoruba were forcibly removed from Africa, some admixture with European and other African ethnic groups occurred in the Americas, creating distinct genetic signatures in diaspora Yoruba populations. Recent admixture, 20th, 21st centuries, modern migration and globalization have increased genetic exchange between Yoruba and other populations. By measuring the precise length of admixture blocks, a technique called admixture decay analysis, researchers could date when these different events occurred, creating a genetic timeline of Yoruba population history. The demographic revolution. Ancient population sizes can be estimated from genetic data using sophisticated mathematical models. These analyses revealed surprising demographic patterns. Bottleneck evidence suggests periodic bottlenecks, sharp population reductions, coinciding with periods of the slavery collapse. The transatlantic slave trade caused a dramatic demographic collapse in Yoruba and other West African populations, removing an estimated 10 to 15 million individuals over 4 centuries. Genetic evidence shows reduced genetic diversity in some Yoruba lineages traceable to this catastrophic period. Recovery and expansion. Following slavery's decline, Yoruba populations experienced rapid recovery and expansion, doubling and tripling in the 19th and 20th centuries, consistent with historical records. Health insights and medical implications. Disease susceptibility. Variants. Studies identified genetic variants affecting susceptibility to diseases, including sickle cell disease, malaria, type 2 diabetes, and certain pharmacogenomics. Yoruba populations show distinct genetic variants affecting how they metabolize medications. This research helps personalize medicine for African populations, often underrepresented in pharmaceutical research. Population reference. Data. Yoruba genomes serve as crucial reference data for distinguishing disease-causing mutations from benign population variants in other African populations, improving diagnostic accuracy across the continent. Implications for human evolution and migration. The Yoruba genetic discoveries illuminate broader patterns of human evolution and migration. African genetic diversity. The research underscores the remarkable genetic diversity within African populations, demonstrating that genetic variation within Africa exceeds variation between Africa and other continents. The out-of-Africa migration. Yoruba ancestry connects to broader West African ancestry that contributed to human populations that eventually reached the Americas during the transatlantic slave trade. Genetic methods can now trace these diasporic connections with precision. Adaptation and selection. Some Yoruba genetic variants show evidence of positive selection, indicating adaptation to West African environments, diets, and disease landscapes. These discoveries help identify genes involved in human adaptation. Population complexity. The research demonstrates that ethnic and linguistic groups are not genetically homogeneous units, but rather complex collections of populations with intricate histories of founding, admixture, and expansion. Methodological innovations. The Yoruba genetic studies pioneered several methodological advances, now applied globally. Statistical phasing. Improved algorithms for determining which genetic variants are inherited together enabled more precise ancestry assignment. Haplotype analysis. Analysis of blocks of inherited genetic variants revealed population history with finer resolution than individual variant analysis. Machine learning integration. Neural networks and other machine learning approaches identified subtle ancestry patterns in large genomic datasets. Integrative analysis. Combining genetics, archaeology, linguistics, and historical records created a more complete understanding than any single discipline alone. Remaining questions and future directions. Despite major breakthroughs, several questions remain. Genetic cultural mismatch. While Yoruba language and culture are clearly related, some genetic patterns don't perfectly align with linguistic classifications, suggesting complex prehistorical interactions. Ancient DNA. Expansion. Sampling ancient DNA from archaeological sites within Yoruba territory would directly illuminate population movements previously inferred only from modern genomes. Selection mapping. Identifying which genes drove adaptive evolution in Yoruba populations remains an active research area. Diaspora genetics. Further study of Yoruba. Diaspora communities, particularly in Brazil, Cuba, and the United States, can illuminate slavery's genetic and demographic impacts. Cultural and social implications. These scientific discoveries carry profound implications beyond genetics. Identity and heritage. Genetic evidence affirms the deep historical roots of Yoruba civilization in West Africa, supporting cultural narratives of ancient origins and continuity. Pan-African connections. Genetic evidence reveals shared ancestry between Yoruba and other African populations, supporting broader Pan-African identity while recognizing distinct histories. The role of African-led research. Importantly, many of these breakthroughs came through increased involvement of African scientists and institutions. Nigerian universities, including the University of Ibadan and Lagos State University, partnered with international collaborators in these genomic studies. This African-centered approach ensures that research serves African communities, respects ethical considerations, and benefits from local knowledge. Conclusion. A complete picture emerges. Ancient West African origins. The Yoruba descend from West African populations with roots stretching back thousands of years population sophistication. The Yoruba developed complex societies with notable demographic capacity. This genetic story, written in DNA and validated through archaeological, linguistic, and historical evidence, confirms what Yoruba oral traditions have long maintained, that they are heirs to an ancient, sophisticated civilization with deep roots in West African soil. Modern genomics has provided scientific validation of cultural memory, transforming genetic data into a narrative of resilience, adaptation, and enduring civilization. As genomic technologies continue advancing and more African populations are studied, similar mysteries will be solved across the science can honor and illuminate the deep human stories written in our DNA.